So I think the topic is pretty clear to our audience. I'm going to talk about reproducible research and how Docker helps us achieve that in bioinformatics. Before I start, I'll just go through the scope of this particular session. I will not talk about or explain in details what is Linux and what are shell commands. I will not talk about in-depth creation of bioinformatics pipelines, and I will also not talk about how to design a bioinformatics experiment. What we would be covering today is the topic of reproducibility in scientific computation, how Docker and Anaconda, these tools, how can they help us achieve it? What are these services? What are these softwares? And why should we use them? And finally, in the end, I will walk you through a live demo on how to work with these systems. To set the premise, I'm going to use a kitchen analogy and talk to you about the four fundamental pieces to achieve reproducibility. So if you keep the image of your kitchen in the back of your head, so we can start with the first couple of pieces which make up the entire environment. So how your kitchen is built, what kind of appliances are inside it, becomes the first set of requirements, and it builds your environment. And this in the technical parallel would equate to the OS or the language that you're using or the software that you would be using for your analysis. The second set of requirements is your ingredients or the raw data. In this case, I have a chicken here. The third set of requirements would be very essential to how you would bring those environment and data together to actually perform your analysis. So in case of kitchen, this would be a cookbook, but in case of your analysis, it would be the code, the documentation, or the user manual. And finally, we have the output for the result in the form of visualizations or graphs. In this case, this would be a fried chicken, right? So this is to set the premise and to tell you what comprises of reproducibility. Next, I will talk about two set of problems that can arise in these fundamental pieces. The first one is a problem that can arise from data or the source of the data, right? So I'm not sure what the background of the audience is, but I'm expecting most of you would be from life sciences. But those who are not, just to give you the context, this particular diagram is taken from the NIH genome website. This is a particular technology that was developed back in 1980s. This is called a microarray. So you have something like a chip, which is more of a glass slide with wells in it. You put in with a known set of genes, you put those in your wells, and you collect samples from a patient. It could be from different conditions, as suggested, normal or tumor patients. You label it with fluorescent dyes, and you let with all the reagents, you let it hybridize in those wells, the sample and your probes. If your samples bind to the given probe in that well, it produces a light of fluorescence, which can be scanned, the intensity of which can be scanned by a particular scanner made for that microarray. And then you are able to quantify those expression results. So ultimately what it does, it helps you to quantify or compute gene expression profiles. Now this methodology, now even though it's becoming obsolete, but back then it was a massive success and everybody wanted to use it. And a lot of papers were released. But a lot of them were also not very reproducible. Other people were redoing the analysis, but were not able to achieve similar results. So in 2010, 2009, 2010, two scientists came in, Bagley and Coombs, and they reanalyzed a set of microarray publications. One of the most prominent ones is a publication from 2006 by Anil Kotri et al. So this particular graph shows you how many publications were analyzed by Bagley and resulted in irreproducible research. And the one I highlighted was a Nature Medicine publication. The key takeaways from this particular paper was they used a cell line, the NCI60 cell line, and they used some 6-7 drugs, and they tried to suggest that by analyzing that the gene expression profiles, they were able to predict if a certain patient sample would be resistant or not resistant to a particular drug. And these were all performed on cancer samples. So when the paper was published, these results got a lot of people really excited at FDA, which regulates a regulating body for drug development and release. But when Bagley and Coombs reanalyzed their data, they were not able to find similar results. One small example I've picked, so one of the drugs they were testing on was 5-fluorouracil. And if you look at this heat map on the left, this is from the Nature Medicine publication, the original publication. And on the right, you will see another heat map from the reanalysis performed by Bagley and Coombs. If you look a little carefully, you would see the data does not overlap. It does not match the results, right? So this was one of the biggest problems during that period, and very similar researches were irreproducible, which not only became an issue in academia, but it had massive implications. Now, obviously, through a period of 3-4 years, with a lot of exchanges, the paper was finally retracted. But the major implication of this was with whatever clinical trials were based on this study, and drugs were given to the patients. It turns out that one of them died, at least according to New York Times 2011 This is the main researcher for that paper, Anil Kotti, who was then a scientist at Duke University. So not only does irreproducible research affect the quality of science and the involved researchers, it can have implications even directly to human health, especially since we're dealing with life sciences research. So this is one of the problems. The second one that I want to talk about, which is more important for today's session, is problems that arise from the environment. Again, to set a basic context, I hope some of you would be aware of what mutations are and what SNPs are. So for a given reference genome, we have a certain arrangement of our ATCC bases. And when we try to compare that with our sample, some of those bases might not exactly match, as you can see from the visual. So these single nucleotide polymorphisms can be very deleterious to human health or any organism's health, but can also be silent mutations. So these biological implications, these can be predicted and detected using certain softwares, algorithms, and tools. So this is a basic workflow. You have a reference genome, you get your NGS data, you do the assembly, you get the assembled genome of the sample, and then you perform variant calling, which essentially picks out these differences between the genomes, and you get your genomic variants. Now, to find out if those genomic variants are actually causing the disease or if they have any bad effect or not, there are certain tools which are based on certain databases. So one of the tools that I was personally using back in 2022 was SNPF. So I took my genomic variants that I was working on, and I tried using this particular tool, which takes in the variants and gives a couple of outputs, like you can see on the screen. So in the column called info, it adds a label called ANN, which stands for annotation, and it tells you if it would have a high impact, low impact, or moderate impact based on databases it internally uses curated from literature. Now, this helps you add biological context to your output and figure out if whatever mutations you've detected in your sample would help you make some relationship, identify some relationship between the phenotype and genotype or not. Now, why am I talking about this tool? Because, like I said, when I was working with my data, I wanted to use this tool. The system I was working with had Java version 8 installed. Now, SNPF is a software written in Java, so it requires a Java runtime environment. And I had Java 8 on my system, but the latest version of the tool was not supporting it. It got installed, but it never ran. It kept giving me an error. And I had no idea what the error meant. So after digging a little bit, I realized that this was a pure case of incompatible dependencies. So SNPF required an updated version of Java, whereas I had an older version. Now, this is a place where Docker comes in, systems like Docker and Conda come in, and help you resolve these sort of issues. So I really did not want to delete the Java 8 because I was using other softwares that were dependent on Java 8. So what I did instead, I installed Docker, and within Docker, I installed an updated version of Java. What it did, it allowed me to run the tool and get whatever output I was trying to get. Now, these two examples might have helped you understand the importance of Docker and probably a little bit about reproducibility. Now, moving on, I would like to highlight the formal definition of reproducibility. So I looked into it a little bit. There's a lot of talk in the scientific community about reproducibility, and different domains have adopted it differently, but the main theme almost remains the same. So this is from the Alan Turing Institute. They have a dedicated wing called the Turing Way, which releases guidelines on how to perform reproducible scientific computation. And they define reproducible research as work that can be independently created from the same data and the same code that the original team used to ultimately get the same output of the same result, as is visible from the illustration on the right, taken from their website. So next, I would give you another example, which is very similar to the SNPF example, but it might help you understand the requirement of systems like Conda and Docker. So consider there's someone who is trying to develop a pipeline to perform a certain kind of research, right? So they have, let's say they have a computer environment and they have these certain softwares installed. So they develop a pipeline and they run it and they get their intended output, right? They share it with the second person. The second person has different versions of the tools installed, or in some cases, some of the dependencies are absent. So what would you expect to happen? Obviously, the pipeline won't run, right? So as expected, you get an error and the pipeline or the analysis does not run successfully. How does Conda or Docker help? We tell it to create an isolated environment with those specific versions of the tools, right? Specific versions of the tools when provided to Conda or Docker, it would create an isolated environment, package it in a way such that it is shareable. So after that, when person one shares this with the other researcher with the same versions of the tools that they used, they got the result, right? So they were not getting it with the versions of the tools were not present for the intent. So this is how Docker or Conda systems like Docker and Conda help you achieve reproducibility. Right. So this is a very one of my favorite publications from PLOS. This came in 2013 and talks about 10 very basic rules on how we can achieve reproducibility in scientific computation. These are the 10 points that they talk about in very brief. I would go through them. I've picked out one of few of the important ones. So the first would be to keep a record on how things were done. Right. So if you remember when we were younger, probably in school, we all used to maintain lab reports, right? Something that looked like this. We used to have an objective, what apparatus we used, what was the procedure. Maybe you all do this even now, maintain lab reports, which is very important because you would ask from your seniors, how did you do it? Can I copy? Even it becomes, I mean, it was not evident when you were doing it, but right now when I'm talking about reproducibility, this makes more sense, right? So we were already trying to do reproducible research, but we were not aware. So this is one of the very basic examples on why it's important to record how things were done. The second one is we should try not to have too many manual interventions because manual errors are very difficult to identify and much more difficult to rectify. Right. So next, I would focus on versioning and public access to your code and your research. So this is one of the widely used web services. This is the main page of GitHub, where you can browse to a lot of open source repositories. You can copy from those, you can modify those as per the requirement of your research. A very similar service is provided by GitLab. It allows you to create your own repositories similarly and share code, collaborate, and also keep track of all the changes you make to your code. Because obviously in given research, you go back and forth a lot of times to make changes, correct your problems, bugs. So these two services are one of the most commonly used ones, GitLab and GitHub. The underlying idea is to keep a track of the changes, not just how things were done, but also the changes that were done to the process. The next couple of points are your intermediate results and how you use them to make your visualizations, right? So whenever you have a graph, whenever you plot a graph, maybe for your publication or for your, let's say, some personal project that you're working on, it's very important to store the data that goes behind that graph. Because if you want to update your plots, if you want to add more annotations or metadata to that graph, it's very important to have the original data that was used to generate that graph. The next one is hierarchical reporting, because I mean, specifically for publications, it is important that you have a way to structure your report in a way that you understand and your peers understand. Eventually, that will help the community. And if you want to make some changes to your own code, maintaining a hierarchical report helps. And finally, the randomness. Why is it important? Because if you're doing some sort of a statistical analysis, it's very important that you account for whatever randomness that analysis can be affected by. With this particular illustration, I have tried to draw a parallel between what Docker does and how we function as complex organisms. So Docker has three very basic parts to it, at least for this particular session. The first one would be your Docker file, which is like an instruction manual or a set of instructions that are required, which is very similar to how genetic information is passed in biological organisms in the form of DNA or RNA, right? So DNA and RNA, we usually consider them as instruction manuals for organisms, right? So once you have the instruction manual, the next job is to create something which performs a particular function. So a Docker file would be used to create a Docker image, very similarly to how mRNAs are translated into proteins, because proteins also perform a particular function, very specific functions. In the same way, Docker images are like proteins, which have a very specific function. But proteins by themselves will not be able to take a form and give you a result, right? Proteins would interact with other proteins and form aggregates, interact with other biomolecules. So in a very similar fashion, Docker images also requires to run as a container to be able to execute its function. So take the example of this lady standing with a laptop. Let's say she's a bioinformatician working on a bioinformatics project. So consider her as a Docker container. Within that Docker container, you have proteins. I mean, Docker image, which is like a protein, which is made up of instructions provided in the Docker file, as provided in RNA, DNA, and RNA to create that protein. I hope this example is clear to you. I've tried to make it as simple as possible. And I've also provided a link that you can go to after this session if you want to know in more depth what these elements of Dockers are and how do they function. With that example done, I would get a little bit technical as to why do we want to learn about Docker and how does it work, the technical architecture of Docker. So on the left, I have kept the architecture for virtual machines. I'll refer to them as VMs. Some of you might be aware of VMs. Some of you have had to install VMs to run certain bioinformatics tools. Maybe the others have not. So essentially, when we're trying to run a particular software, they have different sets of dependencies, right? So VMs are a little older concept, but not obsolete. They are very much required as Docker. But the one disadvantage that VMs have is to run, they would require a full-blown OS, right? So whatever softwares and tools you need to perform a analysis, they require certain libraries and files. So for a VM to be able to do that, it would be like running a second OS on top of your host system. So it makes it very heavy. The file sizes are, the OS is replicated each time you want to create a VM. But this problem is addressed very well by Docker. What it does, it creates a Docker environment or a Docker engine on top of your host OS or your computer, and it allows running particular softwares and tools in isolation without replicating an OS inside it. So to make it more clear, I'll try to explain it in biological terms. So if you think about VMs, they are more like individual cells, right? Cells with their own nucleus. So in this case, the OS would be the nucleus. And if I'm talking about cells, each cell has its own set of nucleus. I'm not talking about RBC. So if you think about VMs in that way as complete individual cells, and in comparison, a Docker container would somewhat be like your cell organelle, right? So multiple cell organelles can exist inside a cell and perform individual separate functions, but they still use the resources of that cell. The resources are shared, yet they function in isolation without affecting the other organelles. They can communicate, but won't take up the resource of the other organelle. So I hope that makes it a little more clear.